Hello again. This is going to be the third video on proteins. In the previous videos we looked at how peptide bonds are formed and broken. In the second video we looked at the levels of structure within the proteins. And I'm going to add in a little extra section here. We're going to look at the different bonds that form in the secondary, tertiary and quaternary structures. Now this information here is probably at the higher level and perhaps not the most necessary for A-level biology. However, if you want to get a better understanding, you can watch this, and it's certainly relevant for our foundation university courses that we run. Okay, so the first of those is the hydrogen bonds. Now I spent a bit of time drawing these in the previous video for the secondary structure. Now the hydrogen bonds form between what we refer to as electronegative oxygens and hydrogens which have a partially positive charge. Okay, the electropositive hydrogens. Now they can form, if we have a look down the bottom here, they can form between a few different atoms in the structure of a protein, and possible combinations are between a OH group, perhaps on the branching part of an R group, and the electronegative oxygen, so electropositive hydrogen to an electronegative oxygen. The electropositive hydrogen from another OH group to an electronegative nitrogen. The electropositive hydrogen in an amine group to an electronegative oxygen, and electropositive hydrogen to an electronegative nitrogen. So like I said, this is a little bit more detailed information that's perhaps not necessary for the A-level, but certainly relevant for the foundation biology. Okay, the importance here then is this idea that they form between partially positive hydrogens and an electronegative oxygen or nitrogen. In the secondary structure, this is within the polypeptide between a carboxyl and amine group. In the tertiary structure and the quaternary structure, these are between the R groups. So electropositive hydrogens and an electronegative oxygen or nitrogen between the different R groups. So obviously the position of an amino acid and the R group present on that amino acid determines whether or not this hydrogen bond can form. And we'll come back to why that's important later on. Ionic bonds, well, they form between the acidic amino acids, or an R group with an acidic R group, if that interacts with a basic, an alkaline R group on another amino acid. So again, these are between the R groups, and they form in the tertiary and the quaternary structures. Okay, so we can see the positive ion in our basic R group there, negative ion in the acidic R group here. They interact together to form the ionic bonds. Disulfide bonds form between cysteines sulfur-containing R groups. So when two cysteine amino acids interact with each other, a disulfide bridge or a disulfide bond are able to form through this oxidation reaction. So the removal of two hydrogen atoms leaves those sulfurs available to bond together to form this disulfide bridge or disulfide bond, and that occurs in the tertiary and the quaternary structure of proteins that contain cysteine. Finally, and this is not necessary to remember for the A level, but you may be interested, some R groups are composed of hydrocarbons only. Okay, now hydrocarbons are non-polar and hydrophobic. So these R groups are hydrocarbons. They are non-polar, and because they're non-polar, they are hydrophobic. Okay, now hydrophobic means they do not interact with water. So to prevent that interaction, the polypeptide chain folds in to essentially to hide these non-polar R groups within the inside of the protein, and that expels water. These interact together, and that can help form the three-dimensional shape. Okay, so this is not on the A-level specification, and in fact it's even unlikely that you'd be asked this on the foundation program as well, but they are certainly present. Okay, so that's what I'm going to do for the types of bonds. I've got one more video to do on what happens if the sequence of amino acids changes in the polypeptide chain. Thanks a lot.